This is video 13, our last video for this part 1 of chapter 10. So this is just something that I found in another anatomy textbook, and it does a really good job of explaining everything we've been talking about in this entire section. So unfortunately, one little thing didn't copy well, and that is our nerve and our presynaptic terminal of the axon as it comes in. So this part right here, numbers 1, 2, and 3, deals with the synapse. So we have talked in our last chapter about how the nerve impulse gets propagated down the axon and reaches the presynaptic terminal. And then we have synaptic transmission using acetylcholine, and we trigger an action potential that travels along the sarcolemma. That goes down into the transverse tubule and causes calcium-gated channels to open on the sarcoplasmic reticulum, which causes a release of calcium into the interior sarcoplasm of the muscle cell, which contains all these myofibrils and sarcomeres. Then we bind the troponin on the thin filament, exposing active binding sites for myosin. And then we have contraction. So power strokes use the ATP, and then we have the movement of those myosin heads like this that causes the sarcomere to shorten. We have calcium active transport pumps that require energy to pump calcium, which is in high concentration now since it was released, to cause a high concentration inside the sarcoplasmic reticulum. And then we go back to relaxation. But it can go back to here if we remain with the cycle. So this does a really nice job of summarizing all that. So just stop, close your eyes for a second, and think of all of the steps that has to occur for you just to move a muscle. So with an action potential, we have to have a graded potential being formed or a signal from the brain that causes an action potential. So we have to have an influx of sodium into the cell that causes depolarization to reach threshold. Then we have to get that message from the brain into the spinal cord, then into a motor neuron to whatever muscle. So if you want to move your pinky muscle of your pinky finger, one of the muscles there, that message has to travel from the brain down to that muscle in your pinky via saltatory conduction the whole way down, which is a good piece if you think about it. It's over a yard. And then we have to go through this entire thing. So we have to go through synaptic transmission. So we have to have that, all those processes. And then we have to have that muscle potential, action potential generated. Calcium has to go down through. We have to have the excitation contraction coupling process go through. And we have to go through this cross bridge cycle just to move your little pinky finger. It's taken us over a month to describe this process that happens in milliseconds. And we're talking about moving one muscle. To pick something up where you have the coordination of all the muscles in your hand and your forearm, that's a lot if you think about it. How many different steps there are and what could go wrong in that process. One little kink in one of these steps can cause a lot of effects. So it's a very elegant system, but it's rather complicated just to move a muscle.